Today I'll be handling the sessions on cloning strategies, especially cloning and expression strategies of the recombinant uh, DNA constructs. First of all, we are going to discuss the steps in gene cloning. In the previous session, we discussed the different terms that is connected with recombinant DNA technology, in which we come across the term recombinant DNA or DNA insert or chimeric gene. All these terms were explained in the previous session. So, before proceeding with the DNA cloning or gene cloning, we need a desired gene. Maybe you are interested in uh, cloning uh, insulin gene, human insulin gene in E. coli, for which you require the source, that is the desired gene. In this case, it is insulin. Or that may be another protein of interest, maybe an anti-cancer protein or an antibiotic gene, whatever. So, a gene of interest should be there for isolation and subsequent incorporation into the recombinant DNA. So, your DNA insert is the desired gene or gene of interest, which you want to procure from some source, isolate it and purify it so that you can insert it in a vector and subsequently that can be cloned. That means the first step in gene cloning is the isolation of the desired gene or the DNA insert, in which case, in this case, we can say that that is the DNA insert or the DNA fragment to be cloned. So the second step, that is the insertion of the isolated gene or the gene of interest in a suitable vector. The vector may be a plasmid or a phage or a cosmid. There are various types of vectors are there. So the second step is the insertion of the gene of interest in a vector. Then comes the introduction of this vector into a suitable host. For example, E. coli cell. And that will act as a host for the amplification of the DNA construct or the multiplication of the DNA construct, and also for the expression of the concerned protein if it is an expression protein. So the host cell, in the case of a insulin gene, which is inserted in a plasmid vector and subsequently transferred into an E. coli, the host is the E. coli cell, in which the protein or will be expressed in large quantities so that it can be purified and used for some commercial applications. So the introduction of this vector or the DNA or the chimeric gene into the host organism is the third step. So that is the transformation step. The next one is the selection of the transformed host cells. So during the process of this cloning, all uh, host cells, maybe there are millions of host cells or billions of host cells in the culture medium that is used for transformation. We can't expect all the 100% of the host E. coli cells were transformed with the concerned DNA construct. Only a portion of that will be transformed, and there will be normal untransformed cells which actually which couldn't uptake that DNA. That will also be present in that culture medium. So from a pool of host cells of a transformed and non-transformed host cells, you need to select the transformed host cells and distinguish it between it from the other untransformed cells. So the selection of the transformed host cells and elimination of the untransformed ones is the selection step. And the next one is the multiplication or expression or integration followed by expression into the of the introduced gene or the desired gene in the host. Usually, when we discussed the previous session, that the purpose or the target of the gene cloning or the DNA recombinant DNA technology is maybe multiplication of the DNA or the DNA of the gene of interest. Multiple copies or large number of copies can be produced by multiplication, or that can be expressed in the host cell. For example, the insulin gene inserted in this manner can be expressed to produce large quantities of the insulin gene, or sometimes you are interested in integrating this gene of interest into the host cell genome, and not in the insulin case, but example in the case of the TI plasmid, which transforms the plant with the Agrobacterium tumefaciens gene. So that TI plasmid, which is incorporated into the Agrobacterium, which is a host cell, that will be used for a subsequent integration into the plant. So that type of transformation is also available. So there, the final step is the target, that is a multiplication of the DNA or the expression of the concerned protein which is coded by the DNA, or the final integration of that desired gene in the host cell genome. So this is the steps that is involved in the gene cloning process, and we will see one by one, and the various strategies that is used for achieving these cloning using different methods that will be discussed in this session. So this diagram or this figure illustrates the basics of the cloning process. Here we have the eukaryotic chromosome which is represented there, and also there is a cloning vector which is a plasmid vector which is represented here. So our cloning process actually it involves the cleavage by restriction enzyme or other methods and separation of the target or the desired gene. So the desired gene can be separated from the eukaryotic chromosome. Say that may be an insulin gene which is obtained from the eukaryotic chromosome. That is the first step. Now we have the cloning vector which is actually cleaved by the restriction enzyme, and the same restriction enzyme that is used for preparing the DNA insert can be used for cleaving these cloning vector. And once it is open, these two things, that is the target gene or the gene of interest and the concerned open plasmid or the vector, that can be ligated together. So the recombinant vector is now created, or the recombinant DNA is now constructed. So the recombinant vector is used for transformation of the host cell. So the next step is first of all, isolation of the desired gene. Now insertion of that gene into the host into the vector. Then finally transformation of that vector, the transformation of that host cell, bacterial host cell, maybe an E. coli, with the recombinant vector. So that is the third step. Then finally the cell propagation will result in multiplication of the DNA construct and also expression of the protein that is coded by that DNA construct. So these are the different steps in the cloning process. So now we come to the cloning strategies. Now by the time we discuss the various steps involved, now we have to see how we can achieve these goals. The initial step is nothing but the preparation of the DNA fragments or the gene of interest, for which you can use various methods. So various methods in combination, any of the method can be used for achieving the first step. For example, DNA fragment preparation, then joining to the vector, then introduction to the host cell, and screening and selection. So this can be categorized in, or this can be layered in 4 levels, 4 stages. The first one is the preparation of the DNA fragment. Then the second one is joining up to the vector. The third one is the introduction to the host cell. And fourth one, selection or screening of the successful transformants. And any of the methods described here can be used for obtaining the DNA fragments. Then subsequently you can proceed to the second step. Restriction endonuclease dissection is a standard procedure that is used for preparing the DNA fragments from any genome, host cell genome. You know, there are in the previous session we discussed the nature of the different types of restriction enzymes that is used for the recombinant DNA preparation, and such type of specific restriction enzymes can be used for cleaving at specific targets or restriction sites in the genome, so that desired gene can be cut off, or it can be cut and extracted from the host cell genome. Another method is mechanical shearing, like sonication or various other mechanical shearing methods can be used for breaking the DNA into random fragments. But specific cutting at specific locations is not possible with the mechanical shearing. But sonication or some other methods of a physical disruption of the chromosome may result in fragmentation of the DNA, and that fragment may contain your gene of interest in some cases, but not always. But if you are looking for complete cloning of a functional gene from start to end, then you may require a more specific method of cloning strategy. Whereas if you are looking for entire genome sequencing and the start and end portion of the gene doesn't matter in that case. In that case, any fragment of the DNA can be cloned. So mechanical shearing will be sufficient for preparation of a genomic library, which we will discuss later. So there are various methods. Anyway, restriction to nucleus digestion is one of the methods which is widely used, which uses specific cleavage at specific restriction sites, whereas mechanical shearing is not. It's a random break that is created by mechanical shearing, which randomly breaks the DNA into different fragments, and each fragment can be cloned into a vector. Then another one is a duplex cDNA synthesis. That is more important when you are looking for cloning a functional gene and you are expecting a functional protein to be produced at the final stage. In that case, you need the complete gene from start to end. That can be achieved only when you are using a cDNA library synthesis. And we will see how cDNA is synthesized in next sessions. Anyway, a duplex cDNA, which is actually a complementary DNA developed from the functional mRNA, and that means that cDNA or complementary DNA developed from this functional mRNA will have the complete sequence of the gene corresponding to the gene, which we can produce a fully functional protein. So in such cases of cloning, you require a duplex cDNA synthesis method for obtaining your DNA fragments or DNA insert of interest. Now, the next one is a direct chemical synthesis. If you are cloning some specific DNA sequences or a specific gene of interest, then even you can synthesize thousands of base pair like the DNA by a chemical synthesis method. A DNA synthesizer machine can be used for a direct chemical synthesis, and nowadays basic techniques like synthetic biology is developed based on that concept, where you only require to give an input into the machine, the sequence of the construct that you want. The machine will synthesize the complete gene of interest or the fragment of interest by pure chemical synthesis. Such type of DNA synthesizing machines are available, and such direct chemical synthesis methods is also another option. And another one is a PCR, where you will amplify the fragment of interest from start to end using specific PCR primers, and PCR based cloning is also widely used. For example, specific gene cloning is widely amplified, and it is used based on PCR based techniques. So these are different methods that is used for a DNA fragment preparation or gene of or isolation of the gene of interest. So that is one of the cloning strategy. Then the second stage, when we come to the second stage, there we have homopolymer tailing, because we are trying to joining this DNA or gene of interest into a vector. So for which you can use a homopolymer tailing. Homopolymer tailing is like adding a poly A or poly T tail at the end of the DNA. Because you can see that all eukaryotic mRNAs is having a poly A tail, and the complementary DNA which is produced from that poly A, all of them will have a poly A tail at the end. Poly A poly T combination will be there. So if your DNA insert that you want to clone, that if that can be joined with a poly A poly T tail, and subsequently it can be joined together specifically during the joining of the vector. So such type of homopolymer tailing, and we will see some of the examples in later slides. So homopolymer tailing is one of the methods in which poly A or poly T or some such specific nucleotide sequences were added at the end of the DNA fragments to be joined, and that complementarity between the polymer tails that is used at the end of the fragments is used for specific joining, and that's actually based on a cohesive terminae is created by the polymer specific polymer tail, a poly A or poly T or poly G, a tail that is specifically attached at the end of the DNA fragment will act as a cohesive end or a sticky end, which will subsequently pair with another complementary pair. For example, a poly T tail will pair with another fragment with a poly A tail, and that sticky end pairing can be used for joining to the vector. Ligation of cohesive terminae. In the previous class, we discussed the restriction digestion and the cohesive terminae or sticky end creation. It is already discussed, and both type of restriction enzymes are there, like the restriction enzymes which creates cohesive or sticky ends, and also restriction enzymes that create blend ends without any sticky ends. And ligation of cohesive terminae is much easier because an echo R1 site with its specific sticky end will always pair with another echo R1 fragment that is created with the same sticky end. So it's a very simple process, a ligation of a cohesive terminae. But when we come to the blend-end ligation, there you require either blend-end ligation enzymes or some other specific methods. So blend-end ligation is also possible because if both the strands are having the same length, and then a blend-end ligation by just like a welding that is required for blend-end ligation. Such type of enzymes are also there. In the previous session, we all while we discussed the different tools or modifications of the cut ends, we discussed about the polymer tailing and also modification of the cohesive terminae, either shortening or extending of the cohesive terminae, and creation of the blend ends by S1 nuclease, etc. were discussed in the previous slides, in the previous class. So you can see that homopolymer tailing, then ligation with the cohesive terminae, ligation with the blend ends, and even addition with the linker molecules. If you are looking for joining fragments created with two different restriction enzymes, you may require a linker molecule or an adapter molecule. So that is another possibility. Then PCR products. All the PCR products will have a specific T extending it, a single nucleotide protrusion or sticky end at the end of all PCR products, and that is one of the features of the Taq polymerase. So that single nucleotide protrusion, which is present at the end of the Taq polymerase, can be also present at the end of all PCR products, can also be used for creation of it can be treated as a sticky end, so that a specific TA pairing can be used for joining to the vector, and that is known as a TA cloning. Anyway, we are not going to the details of the specific methods in this slide. In this slide, we are actually discussing the different strategies or the combination of the strategies that is used for achieving cloning at these 4 different levels. So this is the joining to the vector. Is it clear? Hello? So the third step is introduction to the host cell, where we can use transfection with the recombinant phage DNA or transformation with the recombinant plasmid DNA, or even in vitro packaging into phage codes, transduction with the recombinant phages or plasmids. So there are various methods based on the nature of the vector that is used. You can have different methods of introduction to the host cell. For example, here we have an E. coli as the host, and in which case you can have a plasmid easily integrated into that host cell by transformation, or in some cases, for example, if you are using a phage as the vector, then you may require a transfection based method in which the target host cell is infected with the phage DNA, and subsequently it will amplify in that concerned host cell using its transfection methods. So these are different methods like transfection, transformation, etc., which is used at the third stage. Now, selection or screening is the final step. So after this introduction to the host cell, as we discussed earlier, uh, all the host cells were not transformed because we can't expect so. It's only a random possibility that some of the cells in the medium, some of the host cells in the medium, out of the millions of cells, may uptake the DNA and undergo a successful transformation. Whereas the other ones, untransformed cells, will be still there in the medium, which we want to exclude during the further processing of the of the recombinant DNA constructs. So here you want to use some specific methods to identify, to select and screen the successful transformants with the concerned DNA construct or the gene of interest. So there are such methods like hybridization, PCR-based identification, immunochemical detection, protein-protein interaction, or the protein ligand interaction based identification, functional complementation based identification, gain of function based identification. So there are a lot of different methods, and we can see it in detail. These methods can be discussed later in another class in detail. So the screening and selection of the successful transformants is the final stage, which is the most important part, where unsuccessful transformants were eliminated and successful ones will be picked or handpicked, and that can be used for further use of that successful transformants. So this is the 4 different stages that is described in the cloning step process, and to achieve all these 4 targets or goals, we have many strategies, and any combination of the strategies can be used according to your interest and your resources. Sometimes you may not be be provided with some of the facilities, maybe some chemical restrictions or some other facilities, some other instruments or requirements may not be satisfying your needs. So in that case, you can use any of the combinations which will be which will enable you to achieve this goal. For example, you can use a PCR-based DNA fragmentation, and then you can again go for some ligation of the cohesive termini or a blended ligation, and then you can go for a if your vector is a plasmid vector, then you can go for a transformation with the recombinant plasmid with the E. coli, and subsequently you can use, for example, a PCR-based identification for selection. So any combination of these methods can be used according to your interest and your specific protocol. So the cloning strategy is a most important part, and that the proper strategy is required for cloning of your desired gene. Is it clear? Now you can look into this slide. So the cloning strategies that require generation of the DNA fragments from either genomic DNA or an mRNA, or by polymerase chain reaction or direct chemical synthesis. So various methods can be used for obtaining the DNA fragments. So generation of the fragments from genomic DNA, mRNA, direct chemical synthesis, or polymerase chain reaction. And from genomic DNA, you can use mechanical shearing or restriction digestion. And from mRNA, you can use cDNA synthesis, which gives DNA fragments for cloning. And this also gives the DNA fragments for cloning. Now, it requires methods for joining of the DNA fragments to give a bimolecular recombinant or a concatenameric phage or DNA insert. So that there you require a specific vector for that one, and there are methods which is used for cutting and joining of these fragments, blend and ligation or linkers, adapters, homopolymer tailing. So different types of methods were used for this cloning strategy. So such type of methods were required, and using that one, you can join these fragments into a vector, and that produces a cDNA library or a genomic library which is incorporated with your target gene, and that can be used to isolate the gene or incorporate that gene into some host cell, or express it, or even you can use that gene, cDNA library for DNA sequencing purpose or entire genome sequencing. So anything can be done using such type of cDNA library or a genomic library. Now this is another slide in which the linkers is shown. Yesterday we already discussed the concept of the linkers. Here a 10 base pair 10-mer linker is 10-mer sequence is shown here. This is a linker sequence, and here you have this 10-mer sequence that contains a recognition site for echo R1. So this linker sequence contains recognition site for echo R1. So you can see in DNA ligation, your DNA fragment is now joined with the echo R1 linkers on both sides. The treatment with echo R1 will cleave the linker molecules on both sides, and now a sticky end of the echo R1 is now attached on both sides of the gene of interest. So the construct is then digested with echo R1, which cleaves the linker and to generate protruding 5' termini. So this is one of the methods of addition of the linker molecules, which is attached to the gene of interest or any fragment. This may be a vector, or this may be any blended DNA that you want to clone to an echo R1 site. So once these type of linkers are attached to the blended DNA, it can be easily joined with these echo R1 linkers to other echo R1 DNA fragments. So this is about adapters, use of the adapters. Here we have an example, and this is an example of a BAM-H1 adapter. This is a BAM-H1 adapter, and this is annealed with a single stranded HPA2 linker. So this is another linker molecule. So the BAM-H1 adapter is joined with this one. Then a DNA ligation is used for attaching this molecule on both ends. So this is added to the blended DNA using DNA ligase. The DNA is therefore gains protruding 5' termini without the need for digestion of the restriction enzyme. So without a restriction enzyme, now it is joined with a 5' termini. And the 5' termini of the adapter can be dephosphorylated to prevent self-ligation. Usually dephosphorylation, here you can see that there is no phosphate group, it's an OH, because the phosphate at the 5' end is now removed by dephosphorylation. Yesterday we discussed the dephosphorylation in the modification of the cut ends. That in that step, the phosphorylation of the dephosphorylation process were already discussed. Dephosphorylation of the 5' end is basically used for preventing the self-ligation. So once it is removed, this phosphate group of the 5' is removed from the fragment. Now it is unable to self-ligate or circularize. If it is a vector, the vector is prevented from circularization until the phosphate is replaced. So this type of constructs can be used and it can be attached to the DNA or DNA fragments so that an adapter molecule can be created. This is another one where we have a homopolymer tailing. This is another method in which we have a this is a case where we have the strand is cut with a PST1 enzyme. So this is the PST1 cut. So now we have a poly G attached here. So this is the poly G. A homopolymer of a guanine residue is attached at the end of the 3' end, and both the strands were now attached with a homopolymer tail with a G. And another insert is now, so this is the gene of interest, this is the vector. So the ends of the vector is joined with the homopolymer tail of G, and the insert gene of interest is now joined with the homopolymer tail of a C. So that G and C pairing will result in specific join. So this is the recombinant construct, or the B, or this one. This is the vector, and the DNA insert with poly C on both ends is this one. So this can be joined. This G and C can be paired. This G and this C can be paired. So that results in circularization and successful recombination of that vector and the insert. So this is about the homopolymer tailing. Two complementary tails of poly A and poly C can be used as a homopolymer tail, or you can use poly T and poly A as the tails on both ends of the vector and the DNA insert, so that it will pair and circularize the concerned plasmid, or and so that it will create the recombinant construct. So this is about cloning of the cDNA, in which a double-stranded DNA that is treated with echo-R1 methylase. Echo-R1 methylase is a methylase enzyme that specifically methylates the DNA strands. So once it is methylated, you can add echo-R1 linkers. Echo-R1 linkers were added on both sides. So the echo-R1 linkers, as you know, this contains echo-R1 restriction site. So the double-stranded DNA, or the gene of interest, is now blocked by the methyl groups attached by the echo-R1 methylase. Then subsequently you are attaching the linkers. Then you can digest it with the echo-R1 restriction enzyme. So on both ends you have now the echo-R1 sequences or sequence on both ends. So this can be used for joining with a vector which is cleaved with echo-R1. So if the vector is cleaved or cut open by the echo-R1, this construct can be easily joined, because this will be complementary to the echo-R1 cleavage of the vector. So that will be joined and specifically positioned at that DNA, so that the recombinant DNA can be easily constructed. Let me ask you a question. If anybody can explain me why you are using echo-R1 methylase during this process. Anybody can suggest me how it will be helpful in this cloning process. What is the application of echo-R1 methylase? പറയാമോ? Hello? Hello? ആരെങ്കിലുമൊക്കെ റെസ്പോണ്ട് ചെയ്യൂ. What is the application of the echo-R1 methylase in this process? എല്ലാവരും വിട്ടോ? ഹലോ? ഉണ്ടെങ്കിൽ ഉണ്ടെന്ന് പറ, ഇല്ലെങ്കിൽ ഇല്ല. യു ക്യാൻ ആൻസർ. echo-R1 methylase application. What is the use of echo-R1 methylase? Without that step, what will happen? That's what I'm asking about. Okay, that strand is now methylated. And what's the purpose of methylation? Yesterday we discussed that one. That's why I am asking. Without methylation, what will happen to the DNA? No, without methylation, what will happen to the DNA on the next step? So what will happen when you are applying echo-R1? What will happen? In the next step? No. Actually, after echo-R1 methylation, you are adding echo-R1, right? Linkers. Then you are adding echo-R1, right? You are treating it with echo-R1 or the restriction site, right? Initially, you are adding a coR1. One second. Initially, you are methylating it, then you are adding the linker, then you are diesting it with a coR1. So these points were nothing but they are restriction sites, right? Specific restriction sites. A coR1 methylases will methylate that sites. So without methylation, what will happen? This point will also be cleaved, right? So this is not cleaved by the coR1 because this is methylated. And if it is not methylated, it will be treated as a foreign DNA and that DNA will be cleaved. So you won't get this fragment after adapter. At the end, there will not be any linker, but the strand will be cut short at this in between these 2 points. So instead of adding the linker, your entire DNA strand will be cleaved at all points where echo R1 sites were present, even if it is a self position. Okay, so during the cloning process or the recombinant DNA construction, you don't want your construct to be cleaved, right? This is the double-stranded DNA is your gene of interest, which you want to preserve as such. And there may be restriction sites in between that sequence. Maybe it's a 1000 base pair long DNA sequence or the entire gene. And in between that gene also multiple positions will be there which will be responding to the echo R1. So echo R1 restriction sites can be expected in such a long sequence of the DNA. But the self enzyme will not recognize or will not cleave these points because it is methylated by the self methylase, echo R1 methylase. If this methylase is not methylating this position, what happens? This will be open for cleavage by echo R1. So instead of creating such a fragment, what happens? Without echo R1 methylase, it will be cleaved at these points. Because this is an echo R1 site, this is also an echo R1 site, that will be cleaved. Only if it will not be cleaved and recognized as a self DNA only when it is methylated in its own specific pattern. So that's why you are using echo R1 methylase to methylate the self DNA and distinguish it from the other methylase, other echo R1 sites which is added along with the linker. Okay. So before the linker addition, you require marking of all the echo R1 sites in the self DNA so that points will not be cleaved. Clear there? Okay. So this is some of the genome information available in the NCBI database about various organisms. You can see that eukaryotes almost near 5,000. Maybe it is now over that one. It's the last year statistics. And more than 1,25,000 prokaryotic genomes were now already available in the database, and viruses, plasmids, organelles. So there are a lot of entire genome were already sequenced, and it is now made available in the NCBI database. And that can be used for your recombinant DNA technology purpose, and all the sequencing, entire genome sequencing, and that is done by construction of genomic libraries, and we will see how it is done. So for the sequencing of the entire chromosome of an organism, entire genome of an organism, you require specific strategies, which includes the construction of genomic libraries. Now, isolation of the desired gene, that was the first step. And for the isolation of the desired gene, you can use genomic library, cDNA library, amplification through PCR, and chemical synthesis. And in these sessions, we will discuss genomic library, cDNA library, and amplification through PCR. Chemical synthesis is not our priority right now, but the various items like genomic library construction, cDNA, and amplification through PCR will be discussed in detail. So here, this diagram shows genomic library and cDNA library preparation. So here we have a chromosomal DNA. The chromosomal DNA is having two genes represented here, gene A and gene B. On both sides, we have the same one, gene A and gene B. A eukaryotic gene, as you know, it will have exons and introns and some non-transcribed regions. Okay, so this is the normal representation of a segment of a eukaryotic DNA, where you have a non-translated gene regions of the DNA, which is not actually coding for any genes, and the genes, the region corresponding to the gene, and in that gene itself, you have exons which are joined together to produce a functional mRNA, and the introns which will be spliced off during the splicing process, and they are intervening sequences. So a eukaryotic gene is having non-transcribed DNA, then genes which includes exons and intron regions. Okay. Now, during the construction of a genomic library, this type of entire chromosomal DNA is broken into different fragments by restriction endonuclease digestion, or some other methods like different fragmentation methods can also be used for breaking down the entire chromosomal DNA into smaller fragments. So in this case, you can see that it is randomly broken based on the restriction size that is available, or by mechanical shearing, it will be broken into different components. But the gene A is now represented in 3 fragments. Gene B is also represented in 3 fragments, 2 or 3 fragments. So that means the genomic DNA library is actually not corresponding to any particular gene. A single member of the gene or a single fragment that is created during the genomic library is not an entirely functional gene. It may contain a portion of the gene or a full gene, or a part, or the same gene may be represented in multiple fragments as explained in this case. For example, the gene A is now fragmented into 3 parts, and gene B is now fragmented into different parts. So the entire chromosomal DNA can be broken into multiple or maybe thousands of fragments, and each fragment can be cloned into into a separate vector, and all the collection of for example, the E. coli genome may be broken into 1,000 fragments or 10,000 fragments, and all the 10,000 fragments were individually cloned into a vector, and the collection of the entire clones that carries all the fragments. If it is 1,000 fragments that you have created, you require 1,000 members in the library or 1,000 vectors in the library which carries each and every fragment. So the collection or the sum total of the DNA that is represented in this 1,000 members of the library, genomic library, will contain the entire chromosome. All the fragments will be represented in one or other member of the library. So that type of library is known as a genomic library. Whereas a cDNA library, it is slightly different, where you will the genome or from the chromosomal DNA, the gene is transcribed and the RNA is processed, and the RNA primary transcript is now undergoes splicing and removes the introns. Then functional mRNA is produced. From the functional mRNA, during the reverse transcription process, a complementary DNA is produced. So each member of this cDNA clone corresponds to a functional mRNA, which can be translated into a protein, functional protein. So every member of the clone, cDNA clone, carries a functional gene, even without introns, and that can be used for producing the proteins, or that clone or that member of the library can produce the concerned protein, specific protein from the cDNA library. But the cDNA library cannot be used for say genome sequencing purpose, because it doesn't have non-transcribed regions, and also it doesn't have intron regions, which is also a part of the entire chromosome. So the entire genome is not represented always in case of a cDNA library. Only the genes corresponding, or only the DNA that is corresponding to the functional genes or the mRNA sequences were represented in cDNA library. So that cannot be used for a genome sequencing purpose. Whereas the cDNA library, genomic DNA library members, they represent the sum total of the entire chromosome, entire genome of the organism. And if you can sequence the entire fragments which is created in a cDNA library, then all these fragments, the sequence of all these fragments constitute the total genome of the concerned organism. So this is about genomic library and cDNA library. Now let us see some of the details. The genomic library is a collection of plasmid clones or phage lysates containing recombinant DNA molecules so that the sum total of the DNA inserts in this collection ideally represent the entire chromosome of the considered organism. The genomic DNA libraries are generated by fragmenting the genome and cloning overlapping fragments into vectors. So each and every fragment were cloned, and that members of the library with the complete genome were now known as the genomic cDNA library, and that can be cloned, and even it can be used for sequencing of the entire genome. The exception in this case is due to toxic products of the genes or recombination or slow replication during cloning. So generally we say that the genomic DNA library represents 100% of the entire chromosome, because every fragment is represented in one or other of the vector. But in some cases, some portion may be missing. So you are trying to clone all the fragments in a genomic library, and you say you are breaking it into 1,000 fragments, and you are expecting 1,000 different clones, but you may get only maybe 990. 10 will be missing. Why this 10 will be missing? Because some of the clones may produce toxic products which will prevent its growth. So the clone will not grow because the product will be toxic to the cell. So if the product of that concerned clone is preventing the host cell from growing, then that will not be grown. So it will not be represented in the genomic library. Also, some of the products, slow replication of some of the clones, etc., will result in elimination of that particular clone from the library. So most probably you can see that it will represent the sum total or 100% of the genome. But based on the procedures, maybe if it is producing a toxic product or resulting in a slow replication process, which will gradually eliminate the slow growing clones from the library. So in that case, there is a possibility that some of the fragments may not be represented in the library of the genomic DNA. So this is about the genomic DNA library general concept, and here some of the methods for construction of the genomic library is given here. You can see that the extraction of the total DNA of the organism is the first step. So for preparation of the genomic library, you require to extract the total DNA. Maybe you are trying to sequence the entire genome of a novel bacteria or a plant, whatever. So you have to extract the entire total DNA. Fragmentation of that DNA or chromosomal DNA by mechanical shearing, sonication, or restriction digestion, partial digestion, etc. There are various methods for that. That is the second step. Now, agarose gel electrophoresis of these partial digester or the broken fragments in sucrose or agarose gel electrophoresis or sucrose gradient centrifugation. That's a separation technique which is used for separating these different fragments based on their size, and that is the next step. So once the fragments were created, different based on their size, then that fragments can be introduced into a suitable cloning vector, which is known as a shotgun approach. And one of the example is a lambda vector and cosmids, which can carry up to 23 to 25 kilo bases. That is 25,000 bases can be incorporated or such a vectors. That is compared to the plasmids, which can only carry about a few thousand base pairs. So the size of the fragment that can be carried by a plasmid is much less when you are comparing with the other vectors like lambda vectors or cosmids, which can have a higher capacity. And cloning to a suitable bacterial host is the next step. So the minimum size of the genomic library depends on the complexity of the genome, the length of the insert, or the fragment that is used to be cloned. So the complexity means Maybe you can break the entire DNA into 10,000 fragments and represent it in 10,000 different vectors. So the total number of the members, just like a library shelf which is containing 10,000 books. So you break up your entire DNA sequence or the DNA fragments into 10,000 pieces, and it will be represented in just like a library which carries 10,000 books. Each plasmid or each vector is a book in the library, which carries one of the fragments or one of a portion of the text. Whereas in some other cases, for example, you are breaking the fragments into 10, instead of 10,000, you are breaking them into 1,000 pieces. So you are again loading it in the library. There will be, instead of 10,000 books, there will be only 1,000 books. Initially, the book size is 100 pages, but in this case, the book size will be 1,000 pages. So you can represent the same volume by increasing the size of the capacity of the book. For example, you can represent the same content with 10,000 books with 100 pages, or 1,000 page books, or 1,000 books with 1,000 pages. Both will carry the same content, but the number of books will be different. Similarly, based on the carrying capacity of the vector, a plasmid vector is a small capacity vector which can carry only smaller fragments. So there you require smaller fragments, maybe 1,000 or 10,000 fragments is to be created, and all of them will be represented as an individual member. Whereas if you are using a high capacity vector like lambda phage or cosmid, then you can have a larger capacity book, and that vector carries a larger fragments. So the number of fragments created can be minimized. 1,000 fragment can be created, and it can be distributed into a lambda phage. So the number of books or the number of members in the library will be minimal. So the complexity of the genome or the size of the genome is one of the factors where how much quantity of the genome or the quantity of the base pairs in the genome is one of the factors. So larger genomes require larger number of members in the library, and also then fragments that you are creating, if it is shorter one, you require more numbers of the vectors, or if you are creating larger fragments, then you require only a minimum number of the vectors. So this is how it works. Now, the minimum number of colonies in the genomic DNA library constructed with 20 kb inserts for a 99% probability of all the genome sequences being represented would be 1,157 for E. coli. E. coli genome size is also given there. And for yeast, it is 3,462. Then for Drosophila, it requires about 38,000. And for human genome, it is about 6,90,000. So that much, because the genome size is increasing, and if you are using 20 kb fragments, then the number of members that is required, or the number of specific vectors that is required for representing all these fragments will be more than, or almost it will be 7 lakhs. That much fragments is required for representing the entire human genome as a 20 kb fragments. So this is how the genomic library is prepared. The first genomic libraries were cloned in simple plasmids and phage vectors. More sophisticated vectors have been developed to facilitate the genomic library construction. Then genomic libraries for higher eukaryotes are usually constructed using high capacity vectors, because higher capacity or complex genomes with larger numbers, they require high capacity vectors, because then only you can minimize the number of members in the library. The PCR can also be used as an alternative for genomic DNA cloning. Then long PCR uses a mixture of enzymes to amplify long DNA templates. So this is about genomic DNA library. And this is a strategy that is used for genomic library DNA genomic library preparation. That is the Maniatis et al. Strategy. Here they are using 2 restriction enzymes to digest the DNA at a tetranucleotide restriction site usually. So here we have a high molecular weight eukaryotic DNA, and here is a lambda vector. So the DNA is actually cleaved with two different restriction enzymes, HAE3 and ALU1. Two restriction enzymes, a mixture of the two restriction enzymes is used, and a partial digestion with these two enzymes will create about different fragments, and you can size fractionate, and you screen out 20 kb size fragments from these digest. Eco-R1 methylase is used for methylating the strand. Then you can link it with the Eco-R1 linker. This step is already discussed in previous slide, because you can after methylation of this 20 kb fragment, you can use a blended ligation with the Eco-R1 linker. So your fragment is now joined with the Eco-R1 restriction site on both ends. That linker is attached at both ends. And here we have the lambda vector, and now it is opened with the Eco-R1. So once the Eco-R1 digested lambda vector, that is joined with the or annealed with this cohesive and created by addition of the linker. So that will create a concatenate, or this will be joined. This is the vector, this is the DNA construct, and this can be packaged into a phage particle. So this is how a genomic library is generally prepared using the Maniatis strategy. This is another method, an improved strategy using a single restriction enzyme. In the previous case, we are using two restriction enzymes. SAU3AA, that is another restriction enzyme. Here there is no blended ligation. So it creates a fragments that can be inserted into a lambda vector, lambda replacement vector. This is explained in the Primrose, the textbook of recombinant DNA technology, that is a Primrose, Principles of Gene Manipulation is the textbook from which it is taken. So you can refer to that book. So a polylinker sites is present here. Another polylinker means three different restriction enzymes can be acting on this point. Three different restriction sites were present here. This is another restriction site. This is another restriction site. So this is a specific vector, lambda vector. Lambda EMBL 3A, that is a vector with multiple restriction sites. So this is the chromosomal DNA, genomic DNA. A partial digest with the SAU3A will result in the digestion of this lambda vector and creation of these fragments. And here also we have the phosphatase treatment given here. And finally you have the DNA which is broken, and SAU3I fragments were created from the target DNA also, and these fragments were joined and annealed here, and now subsequently it is packaged into the phage vector. So this is another improved strategy which is using a single restriction enzyme SAU3I for cloning of the desired gene for genomic DNA preparation. Okay, so these two strategies, any of these two strategies can be used, and if you are asked to write some essays on genomic DNA cloning, certainly you can describe the methods, and of course you can explain these strategies with the restriction enzyme digestion of two restriction, a mixture of two restriction enzymes, or an improved method with a single restriction enzyme, and both these strategies can be used for describing the genomic DNA library preparation.